Hello student, welcome to the lecture. In previous lecture we discussed the metal enzyme of copper, that is galactose oxidase. Now in this lecture we are going to discuss the metal enzyme of copper, that is superoxide dismutase. That is SOD. Superoxide dismutase is enzyme that catalyzes the disproportionation of superoxide. It is a byproduct which is produced in the metabolism of oxygen. If it is not regulated, due to this, superoxide causes cell damage. So superoxide dismutase can disproportionate this superoxide into ordinary oxygen and hydrogen peroxide. So let's discuss the structure of superoxide dismutase. If you see in the figure that superoxide dismutase contain two active center, that is the copper and zinc. In that, copper is coordinated with 4 histidine groups and one is a water molecule which is a labile position, and zinc is coordinated or the ligated with the 3 histidine and 1 aspartate residues. In these two active centers, the one that is histidine is a common ligand which is ligated to both active centers. Now let's discuss the structure of superoxide dismutase in detail. In structure, in superoxide dismutase, containing two active centers, sites, which is known as copper and zinc centers. In that, preferentially, copper is ligated with, copper is ligated with 4 histidine and 1 water molecule. Also zinc centers. Zinc is coordinated with 3 nitrogen histidines and the 4th position is occupied by aspartate residues. Now, if you observe in these two reactive centers, histidine is a common ligand which is a bridging ligand in between these two centers. So, in that, one histidine group is ligated both copper and zinc active centers. Zinc 2 plus and copper 2 plus. So if we see the geometry of this respective enzyme, in that the copper is a square pyramidal whereas zinc is a tetrahedral one. So the geometry of enzyme is square pyramidal for copper and tetrahedral for zinc. So here the function of zinc in this Metalloenzyme is it gives a supporting role to these active centers, or zinc play a very supporting role in that SOD, that is superoxide dismutase enzyme. So let's discuss what are the main reaction of superoxide dismutase. As we know that superoxide dismutase can regulate the dismutation of superoxide, which is converted into ordinary oxygen and hydrogen peroxide. So let's discuss. It also acts as an antioxidant in many cell reactions. So let's discuss. As we know that SODs, that is superoxide dismutase, can catalyze the disproportionation of superoxide if it will disproportionate, then it will form that type, that is H2O2. In that way, the SOD, this way, the SOD can convert superoxide into less damaging species. Less damaging species. And if you observe the overall reaction for the superoxide, then it can be written SOD. We use the SOD, that is superoxide dismutase, abbreviation O here. It will be the as like SOD. Copper SOD, then plus H2O2. Means if you observe the side product and here, that is produced into less damaging species here. So these are the overall reaction of the superoxide dismutase. So these two reactions, here there is reduction of copper and oxidation of superoxide. And if we observe the second reaction, then there will be the oxidation of copper and reduction of superoxide. So these are the general reactions which is catalyzed by superoxide dismutase. Instead of this reaction, it will be just inserted the metal. As we know that the types of superoxide is depend upon the cofactors and the protein fold. So these are the general reactions of superoxide dismutase. Now let's discuss the types of superoxide. The protein fold and metal cofactor, there are generally three major types of superoxide dismutase. In that first, that is copper and zinc SOD. Second one, that is Fe and manganese SOD. Third, that is nickel type SOD. So these three types are generally depending upon protein fold and metal cofactor, So these are the three general major types of SOD. Now let's discuss this first type in detail. The first type is that the copper and zinc SOD. So this is this type of structure of this copper and zinc SOD. In that, if you see that again copper and zinc are an active sites, whereas copper is based on square pyramidal, whereas zinc have an tetrahedral geometry. So let's discuss the features or the characteristics. In that, zinc SOD is generally used by eukaryotes cells, by copper zinc SOD is purified by bovine red blood cells as we know that in copper zinc SOD there are 6 ligands including one aspartate group, one of the histidine is visited to both copper and zinc centers. So this is a copper and zinc SOD type. Now let's discuss the Fe and Mn SOD types. Fe, Mn SOD. In that, for Fe SOD, if you observe the Fe SOD, that is a manganese superoxide dismutase, in that actually they are dimers. But sometimes it may be tetramers. The 3D structure of this enzyme manganese and Fe SOD have some arrangements of alpha helix or Same time and arrangement of amino acids. This FeSOD is generally found in prokaryotes. Prokaryotics. Now, the third point is that many bacteria contain FeSOD enzymes. So these are the general features for the iron superoxide dismutase. Let's discuss what's about the manganese superoxide dismutase, M.Sc.II. It is generally found in bacteria as well as human mitochondria. Actually, the structure of this M.Sc.II. contain there are 3 histidines, one aspartate group, residue, and molecule as a ligand. It also found in prokaryotes. So these are the general feature for M.Sc.II. Now let's discuss the third type, that is nickel superoxide dismutase. Nickel SOD. Generally, it's found in prokaryotes. Then it has a hexameric structure. Built from right-handed right-handed 4 helix bundle. Each bundle contains N-terminal hook that chillate और nickel ion. So in that superoxide dismutase, nickel ion is present in the chillate and in the N-terminal hook of the chillate ring. Now the next one, that is the motifs contained in this nickel superoxide dismutase is that motifs means the protein fold. motifs are contained in this superoxide dismutase histidine, cysteine, 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 lysine, and last one, that is a tyrosinase, that is T-argines. So these are the motif sequence which are present in this nickel superoxide dismutase. Now let's discuss what are the main functions of superoxide dismutase. It is a very important part, as we know that the superoxide dismutase is an antioxidant. It will prevent or it will convert the superoxide or dismutase, catalyze the dismutation of superoxide into less harmful species, that is O2, ordinary oxygen, and hydrogen peroxide. So let's discuss the function. So generally the function of SOD, in function of SOD, superoxide dismutase can play a very important role in human, plant, and bacteria also. So regarding with these three criteria, we can discuss the function of superoxide dismutase. So here, that is first is in human, second in plant, and third one is a bacteria. First, SOD second, and SOD third. And SOD third. Here again, SOD first is present in cytoplasm. SOD second is present in mitochondria. SOD third is present in extracellular part. So these three SODs are present in different sites. So actually, SOD first and SOD third contain copper and zinc. Where SOD second diamond, whereas others are tetramers. SOD second contain magnesium. In higher plants, superoxide dismutase as an antioxidant and protect cellular components from being oxidized by reactive oxygen species. These is also known as a reactive oxygen species, that is ROS. Now this reactive oxygen species can ROS, we can shortly written as ROS, can form as a result of drought, injury, herbicides, and fungicides, pesticides, ozone, ozone and plant metabolic activity, pesticides and many plant metabolic activity. So these are the important functions of superoxide dismutase in plant. As we know that the superoxide dismutase can catalyze the disputation of superoxide, which is converted into ordinary oxygen and H2O2, that is superoxide. So it is a less damaging species. Now let's discuss the function of superoxide dismutase in bacteria. In human, white blood cells use enzyme such as a oxidase to generate superoxide and other reactive species and other oxygen species to kill the bacteria which is present in red blood cells. Kill the bacteria. So some bacteria itself produce superoxide to kill the infection or to avoid the infection or to kill being themselves. So here during infection some bacteria produce SOD protect themselves from being killed. Actually, generally when there will generally in human, when there will be the infection, the white blood cells enzyme produce, that is NADPH oxidase, to generate superoxide and other reactive oxygen species to kill the bacteria. So when there will be the infection in human, then this bacteria produce superoxide dismutase to protect themselves to being killed. So this is the important functions of superoxide dismutase. Now we will stop here. In next lecture we are going to discuss the molybdenum metalloenzymes. Thank you.